Okay, so this set of notes is over DNA replication and uh, kind of the steps that happen when DNA needs to replicate before like cell division. So, to kind of think about it and kind of get us back into stuff we learned in November, before a cell divides, mitosis and meiosis, DNA has to be copied. So now that we have a background on the structure of DNA, so the nucleotides, the nitrogenous bases, the double helix, how might that double helix structure make it possible for DNA to go through replication? So go ahead, get your answers into the Ed puzzle box, and then we will continue on. So this unit has quite a bit of terminology, so just be ready for that. How you want to study and prepare for that is really up to you. I know some of you enjoy doing flashcards, some of you like doing Quizlets. Just make sure you know these terms, especially when it comes to the enzymes. So DNA polymerase, helicase, primase, all of these down here are going to be our enzymes. So just make sure you remember what each one's job is. So for our terminology, replication just means that you are making an identical copy of DNA. So we are starting with this strand or these strands of DNA, and we are getting exact copies of those strands. We want them to be literally verbatim. The original strand is the old strand of DNA. It's the like original. So if we kind of take a look here, so if we have our double helix, during replication, we have to take this double helix, basically split it apart so that each side has the original strand, which would be in purple. And then we have to make the complementary strand, which is in green here. Okay, so the original strand is in the purple, which came from the literally the original strand, and then the green is our complementary strand or the daughter strand. Now, we call it a complementary strand because remember that this original strand is going to have the complementary nitrogenous bases on it. And we'll talk more about that in a little bit, but that's why we call it the complementary strand, is because it's not identical to the original strand. It's actually identical to this one's original strand. DNA polymerase is kind of the main star of this show. DNA polymerase is going to be the enzyme that its job is to actually make the complementary nucleotides. So, DNA polymerase makes the new set of DNA using the original strand. So, makes the complementary using the original. Other enzymes that are important for DNA replication include helicase. Helicase's job is to unwind and unzip the strands, the original strands. So it kind of makes it so that instead of having that twisted ladder, the ladder is untwisted. Primase's job is to make these little chunks of RNA, and these little chunks of RNA are called primers. The primers say this is where DNA synthesis is going to start. So primase makes the primer. The primer marks the starting point for DNA synthesis or DNA replication. DNA ligase, this is another enzyme, and its job is to take these little fragments of DNA that are produced and basically fill in the blanks between the fragments. So DNA ligase will come in, and it's a much smaller enzyme than DNA polymerase, and it works a little slower than DNA polymerase, but it will come in and it will fill in these gaps that are produced. And then it will create a covalent bond, pulling together the the sugar phosphate backbone to make sure that everything is sealed together. And then finally, the last enzyme that is a part of this show is telomerase, and telomerase's job is to basically get to the end of DNA. So DNA polymerase, this guy up here, the star, it's a very big enzyme and it needs to attach to all of the nucleotides that it has room for. So if DNA polymerase has room for to read 10 nucleotides at a time, then it needs to be hanging on to all 10. Once it gets to the end of the strand that it's copying and it's down to 9 and then 8 and then 7 nucleotides, it's no longer hanging on properly. And so what happens is DNA polymerase will let go, and then telomerase actually only codes for certain sequences. It's very specialized. It looks for these repeating sequences, but telomerase will come on and it will code for the end or the telomere of the DNA to make sure that nothing is lost. So otherwise, if there's no telomerase, then DNA polymerase basically gets to the telomere of DNA and goes, meh, I'm done. And then that entire chunk of the telomere doesn't get coded. And that is, over time, the telomere will get a little shorter because even telomerase can't code for the entire thing. And that's what accounts for like aging and wrinkles and gray hair is your telomeres getting a little shorter each time. A few more terminology things here, terminology words. The term anti-parallel, basically, I think of it like two sides of a highway. So the anti-parallel means that the strand of DNA runs in a 5 prime, and when I say 5 prime, this is what I mean is the 5 and then this little apostrophe up here, that's the prime. So it runs in a 5 prime to 3 prime direction. Well, the complementary strand, it's not identical to this strand here on the left. The complementary strand is complementary. So to account for that, it still runs in a 5 prime to 3 prime direction, but it's opposite. So again, I kind of think of it like two sides of a highway. If this side of the highway you're traveling south, this side of the highway you're traveling north. Okay, so the replication fork, the leading strand and the lagging strand, these all have a little bit of something to do with each other. So the replication fork, excuse me, and I'm going to use a few different colors here, and that's going to be on purpose. But if we have our two kind of original strands here, so I'm going to make one purple and one yellow. And then our replication fork is this area right here. So this is where helicase is going to unzip the DNA, and we're going to say it's moving in this direction. So it's going to keep unzipping as it goes toward the right side of the screen, so to speak. So if we have our two original strands here, we have our purple and our yellow. This would be the same as having kind of like a purple and a yellow from up here from our original strand. We're going to say that the yellow strand is marked in a 5 prime to 3 prime method. Okay. And then our purple strand, because it's complementary, it's going to have the 3 prime over here on the left and 5 prime on the right. It's complementary to the yellow strand. So DNA polymerase can only move in a 5 prime to 3 prime direction. So it can only work toward the 3 prime. In this case, because helicase is moving the replication fork toward the right, this means that for the leading strand, which in this case would be the yellow strand, so the leading strand is our yellow strand, DNA polymerase is going to move in that 5 prime to 3 prime direction, and it's literally just going to keep following helicase. Just like I think of it like Pac-Man, it's literally just following it. The lagging strand, so in this case the lagging strand would be purple, it has a little bit more complicated process because remember, it can still only go in the 5 prime to 3 prime direction. DNA polymerase can't just switch. So what happens is helicase is moving to the right, but DNA polymerase has to work back to the left. So DNA polymerase will connect to its primer, it'll work backwards, and then it's like, oh shoot, helicase moved forward. And so then it'll kind of jump back and then it'll go this way and then it'll jump back and it'll go this way and it'll jump back and it'll go this way. And when it does that, it makes these little gaps in between where it has to jump to try to catch up to helicase. And these little gaps make the fragments the Okazaki fragments. So, and then that's when ligase comes in and tries to piece everything back together based off of what was left behind from the lagging strand. The semi-conservative relationship, that just means that you have the 5' and 3' strands. The new strands will be complementary to the parent strand or the original strand, and that each new double strand or each new chunk of DNA will be made up of half of the parent strand and half of the complementary or daughter strand. These are kind of your three major steps of DNA replication. All of DNA replication for eukaryotes is going to happen in the nucleus. In bacteria or prokaryotes, it's slightly different because it's just in the cytoplasm and they don't have a nucleus, so they just go through binary fission. But step one, you have DNA is going to unwind at the origin of replication. So that's going to be done via our helicase. So helicase's job is to unwind and make the replication fork. Then once the DNA has been unzipped and it has kind of opened up, now you have the primers coming in. The primers say, okay, this is where we're going to start replication. And then with that, the new bases are going to be made. Oops. The new bases are going to be made via polymerase. So DNA polymerase follows that 5' to 3', makes the continuous leading strand, also makes the chunky lagging strand, those Okazaki fragments. Once it's done, the primers go away. The nucleotides are now in place, and then DNA ligase comes in and seals the deal, seals everything together. And then you could also add one more step after this, and that's the use of telomerase. There we go. Misspelled it there. Telomerase will come in and finish up the telomeres, which is actually on the next slide. Okay. Or sorry, the slide after that. So here is essentially DNA replication using some pictures. So we've got helicase, it unzips the primase. So here's our primase dropping off those primers, those chunks of. So here's our primers here. So the red is primase. Here's our primers. Once the primers say, okay, here's where we're starting, DNA polymerase comes in, starts making the base pairs, and then ligase is going to come in and seal everything up once the DNA polymerase has dropped its the complementary nitrogenous bases. So this is just a little of information about the 5' to 3' strand, and I kind of already demoed that for you guys, that DNA polymerase can only move 5' to 3'. So when it's moving in the opposite direction of helicase, it has to move and then circle back. So it would do this chunk first, and then it would circle back, and then it would do this chunk, and then it would circle back, and then this chunk, and then circle back. And when it does that, it has these little gaps that need to be filled in by DNA ligase. The semi-conservative nature, again, we kind of already talked about that each new bit of DNA, so with the full double helix, the full parental and complementary strand, part of that had to come from the parent DNA, and the other is the complementary strand that is made from DNA polymerase. And so that's the semi-conservative, is that you have this DNA that is extremely old and can be used over and over and over again. But then once this happens, you're actually using the daughter strand, so that DNA kind of moves further and further from the beginning every time the DNA goes through replication. here is the final bit with the telomere. So again, just reiterating what I've already said during this video. So the telomere is just that ending piece of the DNA. It's always a very repetitive sequence of nitrogenous bases. And it doesn't actually code for anything. It's just kind of like, this is the end of the road. This is as far as you've gone. And so, again, once DNA polymerase hits that telomere, it's like, I'm out. I can't do this anymore. Like, I have run out of room. Telomerase, you need to come in and fix this. You need to finish this. So DNA or excuse me, the telomerase comes in and it basically sees this repeating sequence and it's like, okay, this is my time to shine. This is the only code I am actually good for. And so it codes for that little bit. And so that's what's here in these pictures is you have this little bit of overhang. So notice how you have this 3' overhang at the end of the strand. And so telomerase comes in and is like, okay, I got you guys. Like I can move in this direction. And I don't code for very much. And so it just tries to keep that telomere long so that the life of the cell and the life of its DNA can also stay long. All right, so that is it for the notes. As always, if you have any questions, please let me know. Otherwise, we'll do some activities in class to really work on this and drive it home. And especially those kind of confusing parts like the Okazaki fragments and the leading and the lagging. So we'll work on that more in class. But if you have any questions, let me know. Otherwise, I'll see you then.